And let me begin with this topic, because when I was preparing for this session, I got phone from Dr. Vaishali Patil that this is Faculty Development Program, FDP. And a good number of faculties are young one, very new, and they want to learn drug discovery. It's very true, because drug discovery is a fascinating area to develop new drugs for the new diseases, even for old one. For example, we know about corona. We all are suffering and we want to have a medicine for this deadly disease. So everyone is working hard day and night to get a drug for this fatal disease corona. So that's why all of a sudden the process of drug designing and discovery is in limelight. Everybody is looking at us. They are looking at us to give a medicine, a relief from this disease. So this is now a responsibility to deliver to their hopes. And that is why we need someone to do this job. And to this, we need to understand drug discovery. We need to have more people in the field, because we believe that as the number of people in the field increases, more ideas, better ideas, they arrive, they thrive, and ultimately we can achieve our goal. And that's why it is said, if you are doing drug discovery or designing, you are contributing for your society also. So no need to go in the society and do some charity or something like that. Even at home, even at your workplace, you can do quality work. You can do something to minimize their pain by giving good medicines. And therefore, we need to understand the basics of drug discovery, the ideas behind drug discovery, and then learning them. And for this session, I thought the best idea is to have some introductory speech on a topic and then collection of data for different diseases. So this whole session I plan in two parts. The first one is about having idea about drug discovery, and then we'll go to a training session, a small training session on databases. This is because nowadays a lot of information is available. And if we are living in the age of data mining, we are living with data. So this data is actually very useful. And if you are able to use these databases for designing a new drug, that will be more beneficial to each and everyone. So the session is going to be interactive in nature. I love interactive type of sessions where the participants are able to communicate with me. During the session, you can intervene, you can ask your question at any time. There is no problem. You can ask me to emphasize a particular topic. Okay, so I'll be very happy. So I hope so. We'll have a good interaction. We'll have good ideas about drug discovery. Now, friends, let me clear about drug discovery. Drug discovery is a misnomer actually. We cannot do drug discovery because discovery means accidental identification of a new dimension. So it's more accidental and it's not a plan. Instead, we call it as drug designing. Why drug designing is a better word and why it should be used instead of drug discovery. And drug discovery, even though it's very popular word, but throughout my session, I will use the word drug designing. Soon you will understand why drug designing is a better one. Okay, so shall I start my PowerPoint presentation now?
Yes, sir. Sure.
Hello. Emma. Okay. Let me. So let me go for this. Let me check the windows and let me begin it. Pum 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 pum. The slideshow will begin. So I think my slides are visible on your screen now. I can see a handsome man is on the screen and he's looking at the screen. Yeah. So will you please tell me what's actually is visible on your screen so that I can confirm that everything is going perfectly?
Sir, there is an animation writing welcome and there is title written QSAR and hands-on training on data collection.
Yes. Thank you, sir. Thank you. As I said, this is going to be in two parts. The first one is about QSAR. For many people, QSR is old technique, but a lot of advancement is going on. Therefore, we need to understand QSR for drug designing. And later on, now the era is of data, therefore there will be a small hands-on training for data collection. Because I know this is FDP, so we need to have some training session also. The new faculty should learn something from this session. I hope I'll try my best to get you better. Okay. So I always begin with cheaper publicity. Here is my photograph with when I visited Morocco in 2011 with Dr. Tabi Ben Hadda, Dr. Pirilo Gendre from France, Dr. Hafid from Morocco. So during this visit, I learned a lot of things about bioinformatics and physicochemical properties. So this was very good session, a good starting point for me to begin my career. Okay. Later on, in 2016, by mistake, I was in Croatia with Dr. Vesna Rastia. There, as in my introduction, as they told that there was a session or actually a visit for giving training on drug designing to the faculty members and students there. Okay, so these are the different photographs to prove this point and for cheaper publicity. Okay, now let me go ahead. Now we'll start with the word drug. Why we need a drug and what actually drug? Remember, drug is always, as we are the pharmacist, you are pharmacist, I'm chemist. Okay, I'm MSc chemistry, I'm not M.Pharm or B.Pharm. Okay, so we know that drug is a compound which is single active moiety. It should be single, it should not be a mixture of compound. Even though in India, on the counter we are getting a mixture of different drugs, but it's not a good thing. According to WHO, it should be a single moiety, single compound. And anything which we are using diagnosis, prevention, treatment or cure of a disease is called as drug. So if you are developing a molecule which is used for diagnosis of a drug, we can call it as a drug. Okay, we all know about this concept, and we know that majority of drugs are actually coming from natural resources. In different parts of the world, people are using herbs and shrubs to get cured. So these majority of drugs which we are still using, they are somehow derivatives of natural compounds. Only very few molecules are synthetic in nature. Almost we can say it's a 80-20 ratio. Okay, so we have to keep in mind that nature is the good source of getting a good drug molecule or a lead molecule. Starting point can be a natural product, because a good number of natural compounds have optimized properties like with low toxicity, with better metabolism and other properties which we in short call ADMET. ADMET. Absorption, distribution, metabolism, excretion, toxicity. These are almost optimized in case of natural sources. That's why we always try to begin with a natural compound. And whenever we use all these compounds, we call it as chemotherapy. Okay, unfortunately nowadays people are using chemotherapy as a synonymous to cancer, but in real sense, whenever we take any drug, it's chemotherapy. Now let's go ahead and let me clear three different words. Drug discovery. As I said, drug discovery is misnomer. Okay, we cannot discover a drug because we are trying to find out a new therapeutic action of a molecule. So there are huge number of therapeutic actions. So we cannot say by just looking at the structure and the molecule that this drug is good for so and so disease. So discovery is a very tough phenomenon. It's very difficult. For example, in case of penicillin. Now in penicillin, okay, it was accidentally identified as antibacterial. It was not pre-planned. Dr. Fleming just got it that this penicillin molecule is responsible for the antibacterial activity. Likewise, we know about aspirin for a long time, but now recently we came to know that it has good antiplatelet action. So accidentally it was observed. But a better word is drug designing. Drug designing, what we have to do, we have to go for. Drug designing means modifying the molecule, purposely modifying the molecule for better activity and optimized absorption, distribution, metabolism, excretion, and toxicity. The best example is Tamiflu, which was introduced in 2009 for flu. Okay, so another example is Relinza, so many drugs are there. These drugs are in the market because of purposeful designing, having appropriate modification of the structure. So that's why drug designing is a better word. And we also know that there is one more word, drug delivery, because we need to deliver a drug inside our body. So there are different methods for this purpose. Mostly we use gelatin or starch for this thing. So these three different terminologies are synonymously used, but they are different meaning. And they come under the umbrella of drug development. So whenever we use the word drug development, automatically it comprises drug discovery, designing and delivery. So we have to use the correct word when we are talking to someone. I hope the concept is clear right now. Okay, am I audible there?
Yes,
Is
sir.
it clear now?
Yes,
Okay,
yes,
so whenever
you're
you have any question, you can ask me. I request all the participants to share their queries, to doubts, everything, so that it will going to be better for each and everyone. Okay. Hello. Okay, now this is the next slide. This is about conventional procedure for drug discovery or designing. Here we are doing a simple type of research. What we do, we synthesize few molecules, maybe 20 or 30, and then we go for testing. So the first round is synthesis and then testing. Obviously we get some molecule more active, some are moderately and some are less. Then obviously we choose highly active molecules. Again, we prepare more derivatives of the same active molecule. Again, we go for testing. Then we get three types of molecule: highly active, moderately active, and less active. This repeated cycle is actually repeated a number of times till we get a very highly active molecule. This is going to be very costly. So that's why cost for designing a new drug is about 300 million US dollars. This is a very huge amount. Such type of numbers we usually hear about different government scams in India. They are huge in numbers. Okay, so drug designing is not only costly, it needs 10 to 15 years to launch it in market. So it's very long procedure also. Therefore, it is going to consume a lot of time, chemicals, even researchers. Okay, the accident may happen, and so frustrating, so slower. And unfortunately, it has lower success rate. So the conventional procedure is not a good idea to begin with and to get a drug. It's very with low success is causing the problem. That's why we need to smart way. Okay, what we have to go for? We have to go for a smart way. The smart way is on now visible, I think, on the right hand side. You just have the library of molecules. Don't go for hundreds and thousands of molecules. Prepare selective molecules, prepare a library of molecule, then repeat the procedure of selection. Whether I should go for this type of molecule, you'll have to design the rules and then go ahead. So this will speed up the procedure, the cost will be reduced, and then in small time we can get a good molecule. Now we can call it as lead molecule, which has some good desired properties, not a drug, just before a drug. For example, it is in, say for example, in semi-final. So it is molecule in semi-final, it is called a lead molecule. Okay. So let me go ahead if the concept is clear. Now, today we will concentrate on QSAR for doing all these things. Now, this single slide contains each and every aspect associated with QSAR. It involves structure drawing, data collection, data curation, then selecting specific molecules. Then after this, structure drawing, optimization, then followed by again we cure it. Okay, we need to have perfect optimization of molecule. We again go for modeling, then we test the pharmacophore hypothesis. So a lot of things are going on. All these things are associated with QSAR, and you in QSAR do a lot of things. You have to understand not only one molecule. In QSAR we use a good number of molecules. So instead of understanding one molecule, we are emphasizing our similar molecules having similar properties. We call them as congeneric molecules with small difference in their structure, and this small structure difference is highly useful for devising a new drug for a disease. So in QSAR, instead of one molecule, we believe in many. So it's just like democracy, where everybody is ready to give its opinion. Likewise, in QSAR, every molecule is contributing in our understanding. I hope so you'll enjoy this QSAR session. Okay, let me go ahead. Now, a lot of people say that QSAR is theoretical, but let me clarify that QSAR is not theoretical at all. It is based on experimental data. Remember, the very first thing is that QSAR begins with experimental data. So already someone has prepared the molecule. He has carried out the spectral characterization. He has showed the structure, and then he has sent it for experimental biological activity. And he is reporting each and every biological activity in terms of numbers, quantities. It may be IC50, EC50 or LD50, what it may be. But ultimately, we are using the experimental data reported by a scientist where the structures are known. Each and everything has been confirmed. And then finally, we are using the biological data to identify the pharmacophoric features or structural features responsible for the activity or toxicity of a molecule or a series of molecules. That's why it's not going to be theoretical. The good thing is that someone has already done the experimental part. We are actually doing just like management. We are doing post analysis, post synthesis analysis. So this is going to give you rules that what are the structural features governing their biological activity. So ultimately what we are doing, we are comparing the biological activities of one molecule with another. We are trying to find out the reasons for the high and low activity. And ultimately we use validation. We use a lot of statistical techniques to validate our analysis. So statistics is another part. We are using additional part for our rules. Whatever we are doing is always governed by the guidelines developed by OECD. So in short, I can say that the experimental part has been accomplished by someone in advance. Now in QSR, we are actually using the data to identify the reasons for bioactivity of a molecule. So keep in mind, QSR is not theoretical. On the other hand, in drug discovery, another method is very famous, that is called molecular docking. Molecular docking may or may not be theoretical. Okay, if data is available, experimental part has been conducted, then molecular docking is not theoretical. But no experimental data is available, no biological activity is done, then molecular docking is theoretical. But remember, under any condition, QSAR is not theoretical. It is based on experimental data. So anything which is based on experimental data cannot be theoretical. Is it correct? Is it clear, participants? Dear friends?
Yes, sir.
So if you have any question, you can share it with me. Okay, I will have many questions on this session. So this session is actually to clarify your doubts. Okay. Now in QSAR, which is we know now, quantitative structure activity relation is based on simple three different principles. The two are visible on your slides. I'll be very happy to someone to read it, so that let me make the session very interactive in nature. Someone please, so that I'm sure that everything is going smoothly. There is no problem.
Yes, Praveen sir, are you going to make it?
Please.
Quantitative structure activity relationship, similar
Please.
compound behave similarly and activity or property varies with structure.
Oh, thank you, sir. Thank you. Now, why I'm doing all these things? This is because we have to have some principle in hand to work. Now, these two principles given by Dr. Corwin Hensch, that similar compounds behave similar. Obvious, this is going to be obvious thing. If the compounds are similar, they are possible to have similar biological activities. That means they are very similar. They are not going to be vary a lot. Okay. And second thing is activity or property varies with structure. Second thing is about biological activity is changing with structure. So we can say that this biological activity is a function of structure. So if you change the chemical structure, you are going to get a new property, new activity. Ultimately, it is said that activity is actually a final combination of some lipophilic parameters, some steric parameter, and electronic features of the molecule. And there are some unknown factors. So whenever we do QSAR, we always believe that some lipophilic characters are influencing final activity. Some are their steric factors. They are governing our biological activity. And finally, electronic features like dipole moment or other things, they are also there. And finally, something is not in our hand. Let us call it as unknown factors. Okay. And in QSR, what we do, there is simple mathematical correlation. We try to find out between the structure and biological activity. Okay. So as we know now, different parameters are affecting the biological activity, final activity. Therefore, there are multiple variables. The variables could be associated with lipophilicity, maybe steric repulsion, maybe electronic. So all these are brought together, and we try to describe a relation between the biological activity and the structural features. Now these structural features, we call them a molecular descriptors. Descriptor means something used for description. So the word descriptor is coming from description. So we are describing the molecule. That's why the word molecular descriptor is there. Okay. So let me go ahead. Now there are some important steps involved in QSAR. As we all know that the very first thing is experimental data collection. then structural drawing and appropriate 3D optimization, then molecular descriptor calculation and pruning, that means eliminating unwanted molecular descriptors, model building, validation, and finally interpretation. Okay, so if this part is correct, let me go ahead. Okay, let me see. Now, how we can collect the experimental data? Remember, there are different resources for collection of data. Say, for example, you can use research papers. You can collect the data about the structure and about the biological activity, and then you can use it. But in this way, you can collect few hundred molecules only. But nowadays, as I said, we are living in the world of data. Everything is governed by data. We are analyzing it. We are using it. We all know that Google is heavily dependent on data. Whatever the user is doing, Google is analyzing the data and they are trying to give you the suggestions. So whenever we put something in the search bar, we get some suggestions. So this suggestion is because they are analyzing you continuously. They are collecting data for associated with you. Likewise, we are also doing something for chemicals. So there are some famous databases. The very first is Kmbl Database. Okay, and this Kmbl data from UK is very high level data because it is manually curated database of biologically active molecules which have drug-like properties. And in this database, you can have idea about the chemical structures, biological activity, and even you can get genomic data. The link is there. And today we will use how to use Kmbl data for data collection. Okay. Second database which is famous is binding database. We shortly call it as binding DB. Now once again, it is a public web accessible database of measured binding affinities, focusing chiefly on the interactions of proteins considered to be drug targets with small drug-like molecules. So these two databases are very famous and they are public. That means they are freely accessible. You just need to go there, access the web server, and then collect the data for your protein molecule, or even you can use it to identify a target enzyme for your molecule, or you can identify the disease also. That's why these two databases are very famous. Another database which is related to enzyme is Brenda. Okay, in short we call Brenda enzyme database. It is obviously, as the name suggests, you get a lot of information about all the enzymes. Okay, so if you are working particularly on disease, then you will have to identify the main protein behind it. For example, in case of corona, COVID-19, okay, this virus is heavily using main protease, okay, spike S protein and RDRP and different proteins. Okay, those are essential for the survival of COVID-19. So this novel coronavirus is now using main protease. So in Brenda enzyme, you get information about your target enzyme, that is main protease. Then you can collect the different inhibitors available for main protease. You can identify the patterns in the chemical structure of different inhibitors of main protease, and then you can go ahead. So this is the data which we can use in a systematic way to deal with COVID-19. Okay, so let me go ahead. If this is clear, is it clear there, my friends?
Yes, sir.
In case of QSR, a lot of time people get wrong results or their results are very poor, and then they say that QSR is useless. This is because QSR is not useless. We did it wrongly. And if you are doing any mistake at any part, anywhere, you are not going to get the perfect results. So that is why the very first step is collection of appropriate curated data. So today we'll collect the data, but after the data you'll have to curate it. That is called data curation. And after data curation, you'll have to draw the structures. And then after drawing the structure on your computer, you'll have to go for appropriate 3D optimization. Remember, the world is 3-dimensional. It is not 2D. So you need to have correct geometry of the molecule. Otherwise, you are not going to get the perfect results. And whatever the result you will get, they are not perfect. That's why we need 3D optimization. Now let me go ahead. So why this 3D optimization is very important. Now one photograph is now on your screen. Will you please tell me who is this beautiful lady?
Sir, Mona Lisa after she died.
Yes. She's still there. She's waiting for someone to come. Maybe Da Vinci. Okay.
Probably.
So now the problem is that even though you recognize because you have your own natural intelligence. So you correlated the photograph with the photograph of Mona Lisa. Even though she is not looking like Mona Lisa, but some traits, some features are still there. With the help of this similarity, you identified this picture is definitely belonging to Mona Lisa. But this is just like one-dimensional optimization, incorrect optimization of a molecule. Though you will have some idea, but it's not perfect one. Then I used another method for 3D optimization. Even though the method is not perfect, but it is giving better ideas compared to previous one. So it is said one-dimensional optimization or inappropriate optimization is going to give you some result, but those will be very poor, not to the mark. Just like first photograph. Second photograph is much better, but still the beauty is lacking. She is still not so much pretty. She is known for. So once again we are using another method, but again the optimization is not perfect. But if you use perfect optimization method, now in chemistry or chemical structures, the perfect method is DFT, density functional theory. But it's going to be costly around. Even this photograph took a lot of years to get in final shape, final format. Okay, so DFT calculations are there, but those are very hectic, requires a lot of computational power. You need to have high power computers having high RAM, then SSD and all the features. Okay, and then you get the perfect DFT optimized 3D structure. So you'll have to compromise somewhere for time. But if suppose you are going to use correct 3D optimization method, what you will get perfect identification features. So you will be able to identify which feature is present at what distance and how much away from other molecular feature at certain angle. You get more information. You'll have better information about the molecule, and finally you get better description. So as we improve the method of optimization, we are getting better identification, information and description. That's why 3D optimization is very important in QSAR. And if you are not doing it correctly, probably your results are going to be very poor. Okay, let me go ahead. So in case of this molecular descriptor and pruning, we use one-dimensional, two-dimensional and three-dimensional different type of features. This is bound to be there. Even though 3D structure, 3D molecular descriptors are better, but still we use one-dimensional and two-dimensional. This is because they are also representing certain aspect of a molecule. Remember, don't ignore one-dimensional and two-dimensional ideas. These 1D and 2D molecular descriptors are going to give you a lot of idea, even though 3D are the best. But don't forget that a small ant can cause a lot of harm to big elephant. So that is why it is said, when you are doing QSAR, collect as much as data possible with respect to molecules, with respect to target protein and the disease. And then if suppose a lot of information is there, for example, you are collecting the idea about all the charges on the atom, dipole moment, pK, and so much idea. Remember, at present we can have more than 45,000 descriptors for a single molecule. So a lot of information is there, but remember, each and every information available is not going to be knowledgeous, not going to be useful at all. It may be relevant, but sometimes it is reductant, so you'll have to remove it. So removing all these unwanted parameters is called as descriptor pruning. So that's the next step. So what we did, we collected the data, then we drew the structures, optimized them properly, then calculated a good number of descriptors, and then finally we are removing unwanted things. That's done step by step. Then after this, say for example, we have very few molecules in hand and very few molecular descriptors, then we can go for descriptor selection and model building. So after removing unwanted descriptor, now we say that there are few descriptors, okay, and then we build a model. Now remember, my friends, model building is not your job, it's a job of computer. Computer will do each and everything. Different methodologies are available, different free softwares are available. Soon you'll get the list also. So remember, whenever we do QSAR, we don't do anything manually nowadays. Everything is pre-planned. Okay, so not only data collection, but drawing the structure followed by a molecular descriptor, and then pruning, each and everything is done by computer in few minutes only. Because nowadays the computers are very powerful, they do each and everything in few very small time. They require very few steps to be conducted, and the softwares are very smart nowadays. They will give you advice also, and that's why we do it in time, but we have to do it correctly. Different algorithms are available to select appropriate descriptors like stepwise regression, genetic algorithm, mimetic algorithm, particle swarm optimization. So many algorithms are there. Each and every algorithm has its own benefit, and they are there to avoid overfitting, misleading model, and finally have a model with good performance. So this is done by computer by using different algorithms. Let me go ahead. And then different statistical methods are used to build a model. Some examples are on your screen. For example, multiple linear regression is very famous, very old one. But nowadays in multiple linear regression, we can use best multiple linear regression algorithm. Okay, it's a new methodology. Heuristic methods are there, factor analysis there. So many things are there. And if someone is not going for MLR, they can go for the second option, B partial least square. PLS statistical method is there. Once again, remember this PLS is not done by the man or the person manually. The computer will do it on one click. Now, there are different PLS features available there, like genetic partial least squares, factor analysis, PLS, orthogonal signal correction. So many ideas are available. The use of different technology is subject to the person's choice. Okay, so if you want to have higher accuracy, you can change the methodology. You'll have to try for it. Then finally, whatever you do, you'll have to validate it. We are not going for verification. Verification means go for experimental data. Okay, experimental part is there. Unfortunately, we cannot do the verification for each and every molecule. That's why we go for validation of the model. And different methods are available. And again, I repeat this sentence. You are not going to do it. The software, the computer is going to do it. What are the different methods of validation? Or if you're QSR modeling, leave one out, cross validation, leave many out, maybe external validation. You can use PCA, principal component analysis, or you can go for simulated annealing, automated relevance determination. And if you want to avoid all this validation, you can go for Bayesian statistics, or you can go for Gaussian processes because they do not require cross validation. But even though they are bypassing cross validation, you'll have to go for external validation. You'll have to go for proper validation. So even though Bayesian statistic is very famous, but still we are not good at using this Bayesian statistics in QSAR. This is because it requires specific conditions. Whereas the data which we are using in QSAR is related to biology and we need to be certainty. That's why still the people are hesitant in using Bayesian statistics. The old methodology of multiple linear regression, partial least square, they are still very famous. Nowadays, artificial intelligence is coming. We'll talk about this later. So these are the different methods. So as I said you, this artificial intelligence is the new arrival in the industry. Likewise, we have AI, but we are using AI for a long time. Almost in 1960s, we started using artificial intelligence. Remember, artificial intelligence is new for you, but it's very old technique for the people from drug designing. Now remember, from AI, we are not using complete AI, we are using part of it, which is called as machine learning, ML. This machine learning, which is a subset of AI, is used for accessing the data, then analyzing the trends, generating the rules, and then applying the rules. The same thing is done in artificial intelligence. In artificial intelligence, what they do? They collect the data, analyze, prepare the trends, find out pattern, and then they use it. Likewise, in QSAR or in drug designing, we are using it long back in the form of machine learning. We collect the data. Say, as I told you today we are going to collect the data, structured drawing, optimization, everything. Likewise, AI is similar. So even though AI is new for other fields, it is very old for us. We are using it in form of machine learning, but still we are not using AI to a good extent. Why? Why AI is completely failure? Still it is completely failure in drug designing. Now, can you tell me about this photograph? Who is the man in the photograph? Please, dear participants. Can you
Barack
recognize?
Obama. Barack Obama.
Are you sure?
Sir, yes, I'm
Oh,
sure.
so let me tell you. To identify
No.
it, likewise, artificial also identified this Barack Obama. Artificial intelligence and your natural intelligence, but both. artificial intelligence and natural intelligence will identify this as Barack Obama. But let me tell you about in reality.
Ah.
So
Sir,
when, yes.
sir, is my intelligence also artificial? I was thinking it was natural.
No, it depends upon, say, I should not use this word, but it happens. This is because what we do, let me give you the reason now.
Yes.
How AI and natural intelligence work. They try to have some identification of common features. See now the new image. So what we did, we tried to correlate the blurred image with a known image. We try to find out the similarity. If similarity is a good percentage, we definitely correlate it without anything, and then we just give the answer. The AI is doing the same thing. It's trying to find out the answer, trying out to answer on the basis of similarity. Now this concept of similarity is not clear, and that is the problem with AI. Say for example, you and me, each and everyone is similar. We have two eyes, one nose, okay, 2 ears, but at the same time we are different. Even though we are on large scale, on a broad level, we are similar. But when you compare minute things, ultimately, because of the small things, everything changes and we look different. But remember, we look similar also because we have similar traits. Okay, so that is why AI get confused. So in drug designing, even though AI entered, but it is collapsing. Why it is collapsing? The reason is the concept of similarity. Let me give you more examples. Now here is one molecule. Okay. Now, scientists prepared this molecule and it's one more derivative with a change in the structure with of nitrogen only. Now compare these two molecules. They are almost 99% same. Only one of the aromatic carbon has been replaced by nitrogen. Okay. So these are the two different molecules now I can say, because there is change of one atom there. See, simple, only one atom is there. And then when they were treated for troponin I interacting kinase, the IC50 for left hand molecule was found to be 8,000 nanomolar. Okay, the paper is at the bottom, the reference is there. Now, artificial intelligence was applied to predict the activity for the right hand side molecule. And according to AI, the predicted activity is going to be very close to it. That means 7,800. So you'll say, because there is very small difference, it's not going to affect a lot. Hardly there will be a small difference. So AI, like us, like natural intelligence, predicted a very similar value, a very high similarity. But when the right hand side molecule was tested in reality, the experimental IC50 was found to be 80 nanomolar. Can you believe? A single atom caused a dramatic change, where we have 8,000 on one side and one atom is reducing it to 80. So 100 times betterment in the activity just because of one change. So artificial intelligence completely collapsed in this case. So you can say this may be exceptional case. It's not going to be each and every part. Let me give you second molecule, second example. Now see, this is one more example. It was treated for spleen tyrosine kinase. IC50 was found to be 8.8 nanomolar. Very highly active molecule for tyrosine kinase. Now its derivative was prepared again, where only one of the atom was replaced by nitrogen. Why we are going for nitrogen? Nitrogen is very friendly for synthesis and different rings are available. So instead of benzene ring, we use the pyridine ring. So there was only a difference of again a simple nitrogen atom. Now again you can say that, okay, since they are very similar, 99% they are similar, hardly there is going to be any difference between the activity of new molecule. And the same thing happened with AI. AI predicted is to be very close to the previous one. But again in reality, the experimental data show that this IC50 is very less. So once again, almost 150 times betterment in the activity. So even a single atom can cause this dramatic change. And this shows that similarity is a very good concept for AI, but it fails completely, miserably in case of drugs. Even we are using, I think, AI in our mobile phones also. So sometimes it recognizes your face correctly, but it fails sometimes and you are unable to open your mobile. This is because if there is a change, it collapses. It cannot work properly. Likewise, in drug designing, we have. So you can say why this is happening, why this difference in the activity. Now scientists when analyzed, they found that there are some internal features which are responsible for a change in the activity. For example, the left hand molecule, there is clash steric hindrance between the two benzene rings. They are causing a lot of problem. Steric repulsion is there. Whereas when you replace one of the carbon by nitrogen, the intramolecular hydrogen bonding is there and molecule is very stable. And because of this, it is showing a lot of better activity. So this type of things cannot be captured by AI. AI is unable to recognize these types of things, and therefore in drug designing still we are not using AI for drug designing. We are trying to use it, but we know that it has limitation, and therefore it is in infant state. It's beginning. Okay. We are using QSAR. Now QSAR can be used for virtual screening. Remember my friends, previously people used to have molecular docking for virtual screening. Nowadays the new field QSAR is used for virtual screening. And to your surprise, let me tell you, molecular docking has a success rate of only 0.01 to 0.1%. Very poor. Even it is below 1%. So if you target more than 1 lakh molecule, you will hardly get few molecule as a hits. So it's a very poor ratio. Okay. But if you use QSAR for virtual screening, the success rate after experimental evidence is going to be 1 to 40%. Now compare 0.01 to 0.1 versus 1 to 40. So obviously this new idea in QSAR, which is called as QSAR based virtual screening, is changing the field of QSAR. People are now relying more on QSAR for identification of new hits and then getting better molecule as a drug. So the success rate is almost 100 or 10 times as compared to molecular docking. That's why the field of QSAR now is going to have better drugs in smaller time. Let me go ahead. If this concept is clear to you, shall I go ahead? Hello? Hello?
Yes, sir. Yes, sir. Yes, sir.
Okay. Even though I'm a fan of molecular QSAR, but I'm not saying that molecular docking is not good at all. Molecular docking itself is a very good field, even though success rate is low. But when you use simultaneously QSAR and molecular docking, you can get very fine result, and the success rate is even more than 10%. Okay. Now here are some success stories by using QSAR based virtual screening. Very recently, okay, a few years back, Dr. Zhang synthesized only a few molecules. Oh, sorry, they use a data set of more than 3,000 molecules. They tested it for Plasmodium falciparum, that means for malaria. They built a QSAR model, and then on the basis of this result, they chose Cambridge database for finding out new hits. So this huge Cambridge database contains more than 25 million molecules, and out of this, QSAR identified only 176 molecules. These 176 molecules were then subjected to further experimental evidences, and to check the concept correctly, they mixed 42 additional molecules which are actually inactive. So in all, 176 plus 42, so that more than 200 molecules are subjected to the molecular testing, biological testing, and after the testing, they found that the 25 compounds presented very good activity against. Out of this, to assure that whether biological protocol was correct or not, all the 42 molecules which were actually used to check the biological experimental data, all 42 compounds were experimentally found to be inactive. So that means the protocol used by them was correct. Therefore, out of 176, they got only 25 molecules as a sorry for this. Okay. So out of 176 molecule, they got 25 hits. So these were found to be very active against malaria. So this is the victory. The second one is very recent against corona. For corona, Dr. Alves only used a data set of 113 compounds. Out of this 130 compounds, 40 molecules were really found to be active and 73 inactive. And then they carried out QSAR analysis. They built the model. Then they used drug bank database of approved FDA approved drugs. And out of this FDA approved drugs, which are almost, I think, more than 1600, they found 42 hits, 42 compounds. out of 42 compounds they selected on the basis of available biological resources, only in the 11 molecules were tested for experimental validation. And out of this 11 compounds, they found 3 compounds are very, very active against COVID-19. So consider the success rate. Out of 11, 3 molecules are very active for COVID-19. We can call it as almost a success rate of more than 25%. So if the success rate is more than 25%, you can get a drug very easily. That is why I said QSR based virtual screening is giving the molecules in a small time. Okay. So, but let me clarify the disadvantage associated with QSR. Sometimes it can give you false correlation. It could happen. This is because of experimental errors, or maybe this experimental done by you during QSR modeling. So be prepared for false correlation because of the mistakes done by the human. Okay, and therefore you can get very false result. So be prepared. Now, this is the slide showing some free software available for QSR. I'm using all these softwares. Let me clarify this thing. I'm using all these. ACD, ChemSketch, Freeware, PyMol, RDKit, ChemDraw, Avogadro software, OpenBabel, MMTK, PyDescriptor. Now remember, this PyDescriptor has been prepared by me. Okay, so it is available from me on request. We are using Paddle, Build QSR, Vecka, and a lot of R packages are free of cost available. So we are using this different software to perform QSR and QSR based virtual screening. So I hope so this will be very useful to you. This is about database which I'm going to take today. We'll consider this Kmbl database and Enzyme database. As I said earlier, data collection is most important thing in QSR. And here are some different papers published by our group. This is the first. This year we have published few papers. This one is in MDPI, molecules having impact factor more than 4. I think it's about 4.5. Then pharmaceuticals again from MDPI, impact factor 5.82. Then in elsewhere we have publication on COVID-19 in Chemometrics and Intelligent Laboratory System. So this is going to be useful for those who are working in the field of COVID-19. Then we have one more publication in SAR and QSAR. This journal actually accepted our first paper on COVID-19 last year. Again, one more paper on QSAR methodology on human and myristoyl transferase HSNMT inhibitors. So these are the different papers we are trying to use different methodologies for identification of drugs. Okay, so remember that QSR is not magic. We are going to use computers, we are going to use internet for collection of data, then get the correct type of molecule at the end of the QSR modeling, and that will be useful for you. This is because we have a very limited time today. I have selected only first part of the QSR modeling as a training part. That is, we will go to Kmbl database. We'll try to get some molecules for our targeted enzyme or for the molecules. Okay, shall I go ahead now?
Yes, sir.
Okay, so last part is thank you for listening this Masand ki Pasand, but this is not the end. This is the end of very first part, which is about very much theoretical in nature. Theoretical in the sense only you just listen. I got very few questions. Okay, so before starting the second session, I invite all participants to satisfy their queries if they have any doubt, so that when we'll carry out actual experimental part, we'll have this real training, they will get maximum benefit. Even after that session, we'll go for some question answering also. So this is my humble request to all participants to share their questions.
Sir, we are getting a question from Dr. Saurabh Mehta.
Yes, please.
Sir, you can unmute yourself and ask your question.
Please, shall I stop the presentation?
No, sir, probably there is some issue. He's not unmuting himself. Sir, you please continue with the presentation. We will go to him at some other point.
No problem. Okay. I like to have queries from the participants. Okay. So let me start. Just a minute. Let me stop the presentation. I'll have to go for web server. Just a minute.
Saurabh, now you can unmute yourself.
Yes.
Dr. Saurav can unmute himself. He wants
Yes,
to ask.
yes, ma'am. He has unmuted himself.
Yes,
Dr.
sir.
Saurabh, you can ask your question.
This is from DTU. Yeah. So, Dr. Masand, I'm curious to know that if this QSAR based virtual screening is also successful for covalent inhibitors specifically, and in your experience, like if the success rate is any different from the non-covalent inhibitors, or if there is any variation in general, how's the success rate for covalent inhibitors?
So this is a good question, but let me clarify why QSR is more successful. Now, what you are talking about is covalent inhibitors. Covalent inhibitor means there is a formation of covalent bond between the molecule and the enzyme. So that is structure-based drug designing. Now, when we do QSR, it is ligand-based drug designing. We are not considering the covalent features at all. We are using different molecules. So automatically it is independent of whether it is covalent or non-covalent, or it is reversible or irreversible. It is the data, the purity of the data, the experiment data which is available for the large number of molecules is giving you the hint, is giving the success rate. So whether you go for covalent or non-covalent, it's not going to affect your final outcome. QSR, that's why it's more successful. QSR based virtual screening is giving high success rate because it is avoiding a lot of interferences like covalent, non-covalent, reversible, irreversible, and even it is not considering the enzyme at all. It is looking at the molecules only. A lot of molecules are used.
Thank you, sir.
Yes. Shall I begin the second part of the session?
Yes, sir. Please.
Thank you. So let me go to second part of session. Windows are there. And let me go to Google Chrome. So I think once again Google Chrome is visible on your screen.
Yes, sir.
I'm
Yes, sir,
very
it's
good
visible,
at
sir.
yes, I'm very good at black magic, and with the help of black magic I have changed your screen. Though it is black magic, the screen is white. Okay, so I'll put the word Kmbl. So I'll start just like I do it at home. Kmbl EBI. Remember, whenever you want to access Kmbl database, try to use EBI. Okay, that's because they're starting with EBI. Okay, I'll hit enter. And in Google, the very first database is Kmbl database. So there is no trick to identify where the database is located. You simply put Kmbl in your Google Chrome or in Google search bar, and you'll get, I'll open in a new tab. Okay, let it open. So the database will open just like this one. Okay, you'll see this new page. Got it? So whenever you are on the database, you can use anything. Here the search bar is located on the right hand side. You can use anything. You can use the name of the molecule. You can use the name of the disease or the name of the protein, or you can use the Smiley's notation also. Say for example, suppose I'm working on quinolines. So it will try to give you suggestions. Okay, so on the basis of this, you can search ahead. If you want to search specific protein, for example, suppose you are looking for quinoid dihydropyridine reductase or quinolone efflux pump or anything else, you can click on this and you can get some idea. For example, suppose I am working on quinoline. Okay, so suppose quinoline reductase, oxidoreductase is the target enzyme, then I can click on it. So this is the very first idea. But remember, right now we are working on molecules. So what I will do, I will use quinoline and then click on search bar simply. So on clicking on the search tag, I'll get appearance of something new. So it will try to find out different compounds. So now here you can the results. See, there are more than 41,000 compounds with quinoline ring. And these molecules have been tested for 26 enzymes or targets. There are 293 different assay methods have been reported. More than 1400 documents, that means literature data is available. So you can collect all these things at one click. See, for example, here are different derivatives. So if you want to collect a lot of derivatives of quinoline, you can collect it very easily. Okay, but suppose I am interested in my molecule is quinoline derivative and for which disease I can use it. For example, suppose I'm using quinoline. So okay, for which disease I can go for, because the main moiety, the main structure in my molecule is quinoline. I'll simply click on it, double click on it, and it will start giving you the idea. Say so I'll click on the name. As soon as I click on the name, the compound report card will appear. It will give you an idea about for how many different types of diseases it has been tested. So if quinoline has been tested for one type of disease and it has very high activity, and your molecule is also containing quinoline, then the chances are that your molecule is going to be active for the same. So now here you can see a lot of information is there. Okay, and then we'll go to the bottom. So the most important part is the activity chart. So it has been, see for example on left hand side, it has been checked for 21 different types of activities. Okay, so different types of activities have been tested. Now IC50 is there. Okay, so for this molecule quinoline, IC50 is also reported. Say for example, there are only 3 different types of IC50 reported for this molecule. Then it has been 4 different PK value has been reported. So this information is available for a single molecule. Now, for which type of enzyme or protein it can be. Hello. Is there any question there? Hello.
No, sir, it was by mistake. His mic was on. You please carry.
Yes, sorry. Okay, thank you for this. Now target. So for different types of enzyme it has been tested. So how many enzyme it has been tested for? So it has been tested for 9 different enzymes. So now one molecule has been tested for 9 different enzymes. So let us click on enzyme. Now we know it has been tested for which enzyme, because the data has been collected by the scientists working for Kmbl. So here are different proteins for which has been tested. See, the very first target is aldehyde oxidase 1. So quinoline moieties have been tested for aldehyde oxidase. Those have been tested for tyrosine protein kinases, aldehyde oxidase, UDP, glucuronate, transferase 1, and then so many cytochrome P450. So now I have idea about my molecule, because I have synthesized my molecule. What I will do, I will write down the name in the search bar, and then we'll get idea whether my molecule has been tested for specific disease or specific protein. If a preliminary idea is with me, with experimental data, experimental evidence from Kmbl, we are more assured that my molecule is going to be more active in the same, is going to be more active for the same disease or same protein. So the chances of success will definitely increase, because now we know it has been tested and what type of assay has been used. Every information is available on in a single click. So this is one idea, one way to get whether my molecule is going to be active against this or not. So this is one way to use Kmbl database. Another way is to go for advanced search. Remember what we did today. In first part, we use simple word quinoline to identify the different disease or target enzyme for quinoline. But if suppose I want to go for better, then I will go for advanced search. So click on advanced search, a new window will pop up. Let it open. Okay. Yes, it's there. What I'll do, I'll draw my molecule. So you just need a simple practice to draw your molecule. For example, suppose I have benzene. For example, another benzene. Drawing is the best way to get perfect from your molecular data set. And say, for example, I want to increase the number of carbons. Say, for example, carbon is there, a ring is there. Okay, so this type of molecule which we usually synthesize in our laboratories, and then we are happy that my molecule is very active. Say, for example, NH here and here. So this type of heterocyclic molecules are mostly synthesized in India. A lot of molecules are synthesized. And unfortunately in India, what we do, we synthesize the molecule, we go it for testing, and then after getting the result, we get good publication, and then we keep the molecule in our cupboard. Let it die. The molecule will die. Use it somewhere else. Okay, because for biological testing, we need small quantity. Now what I will do, I will change the similarity search. Okay, how much? So there may be more molecules similar to this one. So the similarity search bar is located at the bottom, and by default, the threshold is more than 95%. So if any other molecule has 95% similarity with this drawn molecule, it will be automatically identified. But my suggestion is to start with a similarity search of 60%. My experience for the last many years, I'm using 60%. So this will identify more ideas, more dimension, more biological activity for the same molecule. Just sorry. Sorry for the interruption. Let me click on similarity. Now I'll click on similarity. Okay, after setting the threshold, decreasing it, let it do. So it's very fast. It's working today. Okay. So you can get the Smiley's notation also for the same molecule, which we draw a few seconds back. And now it is searching the similarity of our molecule with others. Let it search. Let me accept each and everything. Okay. Are you getting, my friends, participants? Dear friends, are you getting me?
Yes, sir. It's all coming good.
Yes, as I got instruction from the organizers that there should be some sort of training, some sort of real experimental work. So this is the just beginning. Here we are going to have searching of a database by using structure drawing, or you can use another idea also. So what actually we got? It is showing one record that no other similar molecule is there, nothing. But it has identified this molecule. It is reported in their database. So Kmbl ID is there. We'll double click on it. On double clicking, it will again give us compound report card. So not only by using nomenclature, you can use structure drawing to identify or to locate or to get some diseases or the different enzyme for which it has been utilized. See, for example, once again this one. So the graph is completely different. So here you can see different graphs. So it has been tested for 4 different enzymes. I will click on right hand side. Okay, on the circle. So the molecule, which is an actually imaginary molecule, I thought, but it has been used for cytochrome P450. Okay, so it has been tested for different variants of P450, 11B2, 17A1, and 19A1. So this means this molecule is going to be very good inhibitor of cytochrome P450. So you can test it for this enzyme, and since cytochromes are used in metabolism, it's going to have better metabolism, I think so. Okay, so I'm not going to comment on this, but this is going to give you idea about the different enzymes for which a similar molecule, similar means similar to your compound, has been tested. So if similar compounds have been tested, you can get better success. It's not 100% perfect method, but remember, something is better than nothing. Now we have some idea for which type of disease or enzyme I should go for. So we use 2 different methods. First we use name, then we drew the structure and use it for identification of target enzyme or protein. Now there is one more method. That is, if suppose you are working on a protein, then you can collect the molecule which have been already tested for a protein. Okay, say for example, I'll go for some like as I was working on quinoline. Okay, so I'll go for, sorry, it's not giving the correct idea. Okay, so I'll say for example, quinine 3-monooxygenase or quininoid dihydropteridine reductase. So I'll click on it. Okay, as soon as I'll click on it, it will search for this protein. Okay, for this enzyme. Now it will report how many molecules have been tested. So if I find something data, I can go for, say, yeah, you can see more than 300 molecules have been tested for this enzyme, quininoid dihydropteridine reductase. So you can see different molecules are there. Okay, so the data is available there. Now what I'll do, I'll click on the Kmbl ID, the blue colored. Okay, just click on it, and you get a lot of information about protein. Then it is saying only 4 molecules have been tested. Okay, sorry, 28 molecules have been tested. Okay, their IC50 has been reported. So 28 molecules have been tested. Let me clear it. So these are the 28 molecules which have been tested for quininoid reductase. Okay, so I can collect this data. Their IC50 is available. So I can get some idea whether my protein or which type of molecule should be used. So this is how drug designing is now using the data to get some lead compound or drug compound. This is one way of working. This is though not a good, not perfect, but as I said, you are now some idea about your molecules or about your protein. This will help you to understand in a different way how I can work. This is going to increase your potential. Okay. So this is another way to get the data set. And one more, let me go for Brenda enzyme. So I'll just go for Google search. I'll go for Brenda enzyme. As I said, Brenda enzyme is a database available for different types of enzymes. So on your Google search, just type Brenda enzyme. The enzyme database is available. Just right click and open in a new tab. Let's move to it. So here you have Brenda enzyme. A lot of information about proteins is available. Say for example, I want to search for a specific protein. I will just write down either the name, or you can use PDB ID, or you can use anything. Okay. So you can say, for example, suppose you are working on corona and you are working on main protease. So as soon as we type, it will give idea main protease. Just click on it. It will give you a lot of information about COVID-19 main protease. See the information on EC 3.4.22.69 SARS coronavirus main protease. So the information is available, what are the synonyms and how we can use it. See on left hand side, if suppose you are collecting different inhibitors, so 396 inhibitors are reported for COVID-19 main protease. The information is available on left hand side. Just I will click on inhibitors and the data will appear. The IUPAC name of the inhibitor is available and it has been reported for COVID-19. So this is the information. These are the different molecules, almost 396 tested for COVID-19. So collect this data and use it for identifying the common features to limit the reproduction of COVID-19. Let us stop the spread of COVID-19. Okay. So this is all about Brenda. This is only one part of the Brenda. It's a lot of information you can get. Not only inhibitors, you can get the functional parameters. Those who are working in drug designing and they are developing the new type of assay, this information very useful for you. What is the optimum pH range? What is the appropriate pH range for this protein? So the information is available to you can develop a good assay also. So it is up to you how you use the data. It's not about data, it is how you use it, how you are going to use it for betterment of the human mankind. Okay, so that's all about the today's session. If you have any question, I'll be very happy to have interaction with the different participants. If they have queries, they can ask.
Sir, I think we have a query from Dr. Saurabh Mehta.
Please. Let me stop the presentation, please. Yes.
Yeah, actually, this is Saurabh Mehta speaking. I already asked my question, although,
Okay.
but I am still actually wondering, Dr. Masand, if you could, if I can follow up on the original query I had about the covalent inhibitors. So, based on what you told that this QSAR based virtual screening is basically ligand based method.
Yes.
Then how does it differentiate between the type of inhibitors, whether it can make covalent bond or just non-covalent interactions?
No. Let me clarify. QSR cannot going to tell you whether it's going to be covalent inhibitor or non-covalent inhibitor. It's just going to give you one idea that it's going to be an inhibitor or not an inhibitor. That's the only thing. It cannot give you differentiate between the different types, inhibitor or non-inhibitor. If it is inhibitor, how much will be its ability to inhibit the protein? That much idea you can get it. You cannot get the idea whether it's going to be covalent or non-covalent. That is the limitation of QSAR. Each and every technology has its own limitation. So if it's a limitation, we accept it, because what QSAR is giving us, it's giving us about the type of molecule, whether it is inhibitor or not an inhibitor. If it is inhibitor, what will be the activity, whether it's going to be a high IC50 or low Ki value, whatever it may be, but we can get some idea. So it's not going to give you idea about all these things. It's not a perfect method. No method in the world is perfect.
I agree, sir. Thank you.
Yes.
Hello, sir, we have a question from Dr. Ashwani Mishra.
Please.
Dr. Mishra, you can ask your question.
Hello,
Yes.
sir. Am I audible? Hello.
Yes, you can use any language,
Yes,
English,
sir. Yes, sir.
Hindi.
You order.
No, sir. I'm really happy with your English. It's my second session with you. I had been a member in the training of yours where I learned QSAR, and
Yes.
this is my second time with you. So,
Thank you.
sir, actually, I need a flash of structure similarity what you did in like in the session. Actually, that point of time it was a connection problem in my area. So
Okay. Okay.
if it happened in a fraction of second, you could please do it so that like I could practice it.
Okay. So you want to have a repetition of the
Repetition of
sim
structure similarity
yes,
using database.
this is the one of the strongest area where we are now actually working. In fact, the similarity searching is the basis of artificial intelligence in QSAR. Even though this method is going to give you good idea, but it's not perfect because the limitation is that they have tested one molecule for suppose 10 enzymes only, but one molecule is most of the time active for a lot of enzymes, maybe hundreds and thousands. So we have not tested yet, and therefore they are not in the database. This doesn't mean if your molecule is not having some such this activity, that means your molecule is inactive. It could be it hasn't been tested, and that's what the reason. Okay, so if it is not tested, it is not a database, and therefore we have such limitation. That's why we are expanding the database, and when we expand the database, we get more idea about the molecules, and therefore the idea of similarity searching is more useful when we have huge database. So let me go again for similarity searching by using QSAR. Let me do it. There I was there in, yeah, there it is. So let me go back to Brenda enzyme. Let me go. So is it visible to you?
Yes, sir.
So
Yes,
similarity
sir.
searching is, yes, similarity searching is very interesting area. Now remember, the similarity searching is based on Tanimoto coefficient in Kmbl database, and it is said it's the best technology available for right now for doing similarity searching. Even though Manhattan distance and other criterion are there, but still we are using the old one. Okay, so let's go for the advanced search. Remember, the similarity search is not available on your main web page. It is available from advanced search on the right hand side. So let us click on this. So advanced search. So the new page will appear where you can draw your molecule. If you already smiley's notation, you can paste it. But the best way to work with this is to use the simple drawing methodology. Let us draw the molecule. In few seconds, the molecule will be there, and you can change the sequence of drawing. Okay, it's not the thing that you have to go. Sorry. So let's go for this one. Okay, we'll use the same previous molecule to verify whether we are going correct or not, because we are going to have the same thing. Okay, so this is the same molecule which drew few minutes back. Now look at the bottom. The similarity level is there. Okay, the threshold by default is 95. That means 95% similarity will be considered. If the similarity is 95 or more than that for this molecule, then it will be reported. Else it is going to give you no results. Okay, but let us change it. We'll go to 60%. Okay, so if any molecule has 60% similarity with this molecule, it will be reported. Remember, minimizing the similarity threshold is the best idea to get more. If I'll go to 50%, say for example, instead of 60%, I'll go for 50%, then more molecules will be reported, because any molecule with 50% similarity with this one is going to be reported in the next page. So let us go for 50%. So let's play on similarity. You click. So here the information that 50% threshold had been used to get the results, and any molecule with 50% similarity to the query molecule is now going to be reported here. See. Now you can see here some earlier there were very few molecules reported, but as soon as we change it to 50%, new molecules are there. Look at this molecule. This is very similar to that one. And the level of similarity also reported. Here it is 57.57% similar to our query molecule, this molecule. The second one is 52.77. The third one has a similarity level of 51.35. So these molecules are different levels of similarity. So if you change the threshold, you will get the change in the results. But the recommended value is 60%. At least your molecule should have similarity of 60% to get some biological activity. So here, suppose I'm interested in this molecule because this is very similar, almost 57.57. Just click on it and you will get the idea that this molecule has been tested for different types of biological activities. You scroll down to the bottom and right inside this last circle, right inside. Okay, this is very important. This pie chart is most important. We'll click on it and then it will now bring us on new page for which type of protein is used. So now we get a more identification. This has been tested for proteasome macropene subunit MB1. So this molecule has been tested. So likewise, your molecule has more than 50% similarity. It can be good against this protein also. This is how we carry out similarity searching. I hope so. This
Is this more similar to scaffold hopping? What we do?
Scaffold hopping is a similar but different. In scaffold hopping, what we do, we change the position of different scaffolds or groups. Okay, that will give you numerous derivatives. It is just like finding a similar molecule. Similarity searching is going on, and similarity searching is nowadays used for identifying the disease for your molecule or enzyme for your molecule. Scaffold hopping is just putting one part of molecule to another part, creating new derivatives. Okay, that's a different area, and that's used in virtual screening also. Nowadays they are using scaffold hopping for creating new derivative of most active molecule and then identifying which one is going to be very active. That's a different area.
Thank you very much, sir.
Yes,
and nice to see you again, sir.
Thank you. So let me stop this presentation and come back to the main screen. Yes, I'll be very happy to more questions from the participants. I know they have queries.
We have a question from Mr. Paratpar Sarkar.
Please, yes.
You can unmute yourself and you can ask your question. Hello.
Yes. Please.
Hello.
Yes, please.
Am I audible, sir?
Yes.
Yes, sir. Sir, I just want to, I have a query, small query, sir. The molecules which are there in the Kmbl database, are they all experimentally proven data?
Yes. This
All.
is what actually, yes. All molecules have been tested. First, they have been experimentally synthesized. They are not imaginary molecules. They have been synthesized. Their structure have been established perfectly by using different structural verification method like spectra, etc. Okay, and then they were tested for biological activity for different proteins or diseases. They have been prepared, synthesized, and after that tested also. Everything is experimental. That is what I actually today told. QSAR or this field is not theoretical at all.
Okay. So
They have
that
been
means
tested.
whatever the molecule you have to first synthesize the molecule and then you want to know that whether that molecule which you have synthesized is how much similar to the molecule which is there in the Kmbl database. That's all.
Let me tell you. At home, you can draw some imaginary molecules. Okay, suppose you want to synthesize so and so molecule. So before its synthesis, say right now the molecule is on the paper. So you just draw it on Kmbl, try to find out the similarity. Then you can get idea about your molecule, whether this virtual imaginary molecule is similar to any other molecule or which molecule has been already tested. And if you get good similarity, you can go for that type of reported activity, biological activity, or for protein.
Be synthesized, sir.
It can be. Then you'll have to synthesize. If suppose you get high similarity of your molecule with a reported molecule, okay, then you are sure that this imaginary molecule is going to be active again for so and so disease. Now you have some idea. Now go for laboratory synthesis. then go for the testing. During synthesis, this idea will help you. During synthesis, now you know my molecule is going to be active against so and so disease because I checked it on Kmbl database.
And what is it different from pharmacomodeling, sir?
Obviously, it is very different. Pharmacophore modeling is not going to tell you negative aspects of your molecule, which features are responsible for decreasing the activity. But this is going to tell you which features are responsible. Similarity searching will give you more dimensions of your molecule. Remember, in QSR, we identify features. In someone please mute. I'm not getting. Okay, this is a lot of noise there. Please, it's a humble request to mute yourself. See, remember, QSR is the best technique because it gives you idea about the features responsible for increasing the activity and decreasing the activity, both. All other techniques are going to tell you which features are good for activity. They are not going to give you idea about which features are going to decrease the activity. That is the basic difference between QSR and docking and pharmacophore modeling. Similarity searching will give you only positive aspects, which features are responsible for increasing the activity. Okay. Hello. Hello.
Yes, sir. Yes, sir. Yes, sir.
You got my point?
Yes. He's probably on hold now.
Maybe.
Okay.
Sir, can I ask a question?
Yes, please.
Sir, actually, we people, what we do it, we do the extraction and isolation of the plant extracts, and
Yes.
we automatically we get some compounds. We do it by IR, NMR, and
Yes.
using LCMS we get the particular compound.
Yes.
So can we use this Brenda for checking that whether our compound, before going in the pharmacological activity,
Yes.
can we find out the basically using Brenda or maybe using Kmbl, can we find out the targets for that particular isolated compound?
Yes, this is what actually this session is all for. You are very true. You just need to have correct structure of molecule. Okay. If it is completely established, you can use it to identify the disease. Okay. And the higher success rate will be there.
So then we may need your help also.
Definitely, you are welcome. You are definitely welcome. Definitely.
Fine. Thank you, sir.
I think you will now. You are more now happy to have some idea that we can do it in this way also.
Yes, definitely.
This will bring. I can imagine a smile on your face. I can imagine.
Yes, sir. Your presentation was highly informative. Lot of information was there.
Thank you. Thank you for the encouragement. Any other question? So this session was planned for more question. Okay, for interaction, not for one-sided learning or one-sided. It should be, because whenever we are in FDP, it is faculty development program. It's a development. Let us share the ideas to develop each other. You may ask some tricky questions to me. This will encourage me to read more literature, try to identify the new answers. Okay, so this is how I will develop. I request you to share your queries, please.
So I guess there are no more queries. We're not getting any queries at this point of time, sir.
Okay, it's okay. Hello, are you getting
yes,
me?
Sir. Yes, sir. Yes, sir.
This is my idea. After few hours, I will definitely send the whole PowerPoint presentation to Dr. Vaishali Patel, and I request her to share it with the participants who are interested in getting it. They can ask Dr. Vaishali Patel to get it. Okay, I'll send it to her via WhatsApp. Okay, I hope this will be useful to them.
Thank you so much for that.
Thank you. Yes, thank you.
So if there are no more questions, we can proceed, Himanshu sir.
Okay. So thank you so much, sir, for such an informative and interactive session. And we hope to see you soon in some other programs.
Definitely.
Right, sir. So, and with the permission of the chair, ma'am, you can give your closing remarks, and we will present Dr. Vijay with our certificate of gratitude.
Sorry for interruption. I forgot one thing. I'm very poor at this thing, and very sorry for this. It's called acknowledgement. It's our Indian tradition to give dhanyavad. I'm very poor. I'm very sorry for this. So let me acknowledge for inviting me for this session where I got good number of participants. I got interaction with the participants. They asked their question. I liked it. And if they want to contact me, they can. I'm also thankful to Dr. Vaishali Patil for specially inviting me for this event. It's a nice platform. I learned a lot of things from this because this is the first time I'm using Microsoft Teams. And today they gave me a small training, which was very useful. Thank you for this training also. I'm thankful to all the person involved in the success of this event. Okay, for inviting me, doing a lot of jobs, and my best wishes to them for such a successful event. Thank you. Thank you again.
Thank you.
Thank you so much. Thank you so much, sir. And we are really happy to have your such kind of enthusiastic and informative, along with the hands-on training experience, all the knowledge sharing session. And we do look forward to see you in the coming time again. And the topics which we have selected for the presentation, that is the need of the hour. QSAR is having the wider boundaries. It is not only for the chemistry personnel, it is having a wider applications. We are going through that in the previous sessions also, like QSAR for toxicology, QSAR for medicinal chemistry purposes. Apart from that, if you will be having all the theoretical knowledge, you can go it for using for nanotechnology, and even for environmental purposes, also environmental toxicology purposes. So sir has given us a window, and it's now up to the participants that how we could explore it. And thanks for the hands-on experiment for handling the databases. So definitely the participants are going to be benefited with your knowledge sharing session, sir. And I will like to ask the team members to present the token of appreciation and an honor from our side for you, sir.
Thank you.
Please receive it, sir. Please display the certificate. So this is on behalf of KIET School of Pharmacy and from the entire organizing committee.
Thank you, madam. Thank you.
Thank you so much, sir. So, see you again in the future. Thank you, sir.
Thank you.
Thank you so much, sir. Thank you. So, ma'am, shall we now break the session?
For the lunch break.
Okay. So, dear participants, now we will break for lunch, and we will join here again at 2:30. Yes.